I think you addressed Manisha's question as well in that. So I think you may have hit a couple of birds with one stone, as they say. Lena, I was hoping that you could give us sort of your general, and I know that there are myriad changes or factors, I suppose, facets, but generally speaking, I was hoping you could give us your perspective on at what points in the development progression it's most important to stop and conduct these analytics exercises.
I think there are a lot of steps where it need to be included. So definitely you need to include it, like I already told, all the analytic stuff in the development on a pool as well as clone level. But also in the USP part, you need to include all the analytics you require for having the CQAs present for your product finally. And this also need to be implemented finally when doing tech transfer, when doing production. So it's really necessary to have the broad panel of analytics already included on mostly all steps of the early development and also in the transfer of the process. And that for that, it's really important to have that defined well. I think that was also addressed already in some of the questions before, that it's so important to have that in an early stage defined on a well way, what are the critical parameters which we need to have a look on. We need to look on also when doing the scale up. We mentioned also in the last answers of the question, so we will get definitely other stress parameter when doing the scale up. We will have other shear stress, we will have other mixing rates finally present, so other gradients in the bioreactor which can stress the molecule, which can stress the process itself, and that I want to add here, Arne.
Mm-hmm. Very good. Meinhard, any additions to that?
Yeah, I think maybe one addition here is, so in the oncology field, just what I see here, it's always, I think, when you start with a program, hard to predict in which indication you will later really then go. Therefore, this prediction of a commercial demand is always quite hard, that at least in particular to have it ready already early once you start with the R&D process. But you have to build, of course, some predictions so that you can also, let's say, address the problem on what titers or what amounts you will later end up for a commercial process.
Yeah.
Matt, just to add to that, I think the key, I think Lena mentioned too, understanding the mechanism of action of the molecule, understanding the, of course, the structures, primary, secondary, and so forth. But that is the key, right? How is the molecule set up for both potency-wise as well as any other functionality? So if you, just as an example, if you have a molecule, fusion protein, for example, if a particular portion of the molecule, the Fc domain, is engineered to do certain functionality, and then if the potency has multiple binding efficiencies that need to be measured, so that will define some of the CQAs, and therefore, I would say at least it will give you pointers to Meinhard made the point about what are the key analytics that I need to develop, because it is sort of a parallel process. When you are coming up with a molecule, you don't have all the test methods and the analytics as well. So you're also developing those. So you can't have all of the analytics, and you will be like, as part of the drug development, you may add some, you may remove, whatever, but so kind of figuring out based on the mechanism of action and the functionality of the molecule, what are some of the key ones? There is some standard ones, but what are the main ones, the key ones that really define the molecule? I think if you have that level of understanding, then you can kind of focus on those analytics early stage and then keep adding as you go through the development.